Okay, well, let's get started. Welcome, everyone. This is the Dangerous Idea Seminar series where we talk about new ideas. Doesn't matter what stage they are of the development or fully developed ideas, or you want to present findings, results. I'm Bhutish Jani. I'm from School of Health and Wellbeing. I'm an academic GP. And the idea is to have a half an hour seminar with 15 to 20 minutes of presentation followed by question and answer. So our speaker today is Mila Colados Rodriguez from School of Infection and Immunity, Center for Virus Research. And Mila is going to talk about relationship between herpes virus infection, chronic infections, and degenerative diseases. So without further delay, I'll pass it on to Mila. Thank you.
Hello, thanks for giving me a spot to give a talk. I'd like to clarify that the ideas are my own visions. So I work here with herpes viruses, but they don't have a lot to do with what I present today. They may have more to do in the future, but anyway, I think it's an interesting topic for discussion. So yeah, let's go ahead. Can you see well the presentation? Yes, okay, so then let's go. Hello. Oh. Okay, it changed in my computer, but I don't see it changing there. Let me see what happened.
Yeah, the slides are not changing.
Yeah, they do change in my computer. Let me stop sharing and see what happened.
Okay.
It's very strange. Okay, I'm gonna close that. It was working before. Now it's not sharing.
No,
You don't see that, right?
no,
Okay.
I'm not seeing anything.
Now it's changing but not being shared.
Okay, we can see, I can just about see that you started screen sharing.
Okay.
Yeah.
Let's see if it passes, because I have a different screen on my computer than that one, so I don't know why. Let's see if it moves. Yes, it's clicking. So I just wanted to show quickly the structure of herpes virus, which is the one I work with. I usually use an egg because it's easy to understand. So they got an envelope, which is the shell of the virus, the tegument, which will be equivalent to the white of the virus. And then they got the capsid, which will be equivalent to the yolk. All of those are proteinaceous structure, except the shell, or the envelope, which will be lipidic. And inside there is the information of the virus. So in the case of chickens, of eggs, chicks will have the information to make more chicks. And in the case of virus, is the double-stranded DNA, which has the information to make more copies. Those are big viruses. They are, just for the herpes simplex, is 152 kilobases. So they are large pathogens, but they are very tiny as well in size. This is how they look in reality. And they kind of have a plan for invading our cells, which is making more viruses. So they get in the cells through different routes. They travel through the cytoplasm, and then they release their genome. It's very compact, but then it decompacts over time, circularize, and make more copies to generate more videos. Some of them are empty, others contain the viral information. So this is what is known the lytic cycle of the virus, but there is also the latent one, which happens in memory cells, either neurons or B cells, the immune cells that make antibodies, and is to chromatinase and stay in the cell latent until there is a stress and it reactivates to make more viruses. So those events the work in the lab and working now, that's how we observe it. We use the confocal and we image the cell nucleus, and we can see individual genomes inside of structures called PML nuclear bodies, which are in charge of what's called the epigenetic information of the cell. So it can change what is expressed in the cell, and also produce defenses. But what the virus does is to disrupt them with one of the proteins, and that's how it gets away. So it blocks the cell from defending itself against invasion. As you can see, there are many shapes the virus adopts inside of the cell, and that makes a problem for the cell to be able to detect that virus just in one cycle. And also is a problem for later on, because you see the virus has its own proteins to counteract all the defenses that the cell has. And in many cases, the place where the virus replicates, which is the nucleus, is not even taken into account in reviews. This more recent already takes into account ICP0. This is the viral protein that can wipe out all the epigenetics of the cell and get rid of the defenses. So here are all the targets of the cell targets. You don't have to be able to see them, just to show that there are many cells, many proteins affected by the viral protein ICP0. It's a herpes protein, but all the other viruses have orthologs. And this is what my point, that all the efforts of the cell and of the researcher are focused on proteins. So I want to make you think with me whether it would be an effective antiviral inhibiting each of those host proteins, or inhibiting all the 10 known viral proteins just for the case of herpes. So we are being focused on virus proteins. But the thing is that, sorry, I can't see my other slide though. We got not only herpes simplex, which it causes cold and genital sores, dermatitis, blindness, even fatal encephalitis of newborns, possibly Alzheimer. But we got other viruses. There is herpes zoster, which is a neurotropic virus, as herpes simplex, which cause chickenpox, shingles, and blindness, as well as viral meningitis. Cytomegalovirus, which cause birth defects and hearing loss, as well as a lot of problems during organ transplantation, because they are latent, and then when there is a stress or the immune system is repressed to undergo organ transplantation, those viruses are prone to reactivate and kill the patient. Then we got Epstein-Barr, which is the causative agent of sclerosis multiple and infectious mononucleosis, as well as cancer and other proposed diseases. And then lesser known, which is the human beta herpes virus, which causes the roseola or the fever of 3 days. It's a kind of rash the babies get at 3 days. And it's the only one that can integrate in your genome, in the telomerase specifically. And the last one is the Kaposi sarcoma herpes viruses. So all of herpes viruses are known to infect people, and actually there is one of those at least in each of us. That means that by the time we are adults, we have been infected and still have latent at least one of those herpes viruses. And there are studies I want to say first that there is no cure and it's asymptomatic. So many times we have it, but we don't know it, and we still keep shedding and transmitting them without knowing that. So those viruses tend to infect us more over time. That means that the older we get, the more chances are that we have a herpes infection. This is only herpes simplex, but there are that's the same for all the other herpes I have shown you. And it's a pity that it causes fatal neonatal encephalitis That's about over 10,000 cases of years. Those are a medium-sized town population of babies that are affected by this virus. Many of them die, and others are left with sequelae over time, and there is no treatment. They are loaded with ineffective antivirals, and yeah, it's a pity worldwide, especially in certain regions of the globe. And there is no cure because we have been focusing on targeting viral proteins one by one, while the virus has many proteins to disable our cells. And also we are targeting on host proteins, like a lot of targets are epigenetic factors, and they are factors that our cell need to function. So the herpes viruses have been not only fouling the cell, but also scientists by making them think that targeting proteins is the solution. So instead of proteins, let's see if that works. I propose to focus on the virus genetic information because I consider that if you remove the information to make more viruses, you stop making them. They are gone. There is a cure. So a good strategy would be that, to eliminate the virus information or target the virus cell interface. Because if you target any components of the cells need them to function. So it wouldn't be like a very clever idea. So this is a publication that has shown that chronic diseases like multiple sclerosis are actually caused by a herpes virus. In this case, we got Epstein-Barr, and it was clearly shown that Epstein-Barr increased the chance of having multiple sclerosis in 32-fold changes. That means that if you got this virus, you have higher chances of developing multiple sclerosis. And there was no multiple sclerosis without the virus. So it's an underlying cause, a part of your genetic makeup, of course. So how can we eliminate the viral information? So our body got proteins which are called DNases. They degrade nucleic acids. Of course, they are very regulated because we don't want to degrade our own DNA. And when they are mutated, they causes autoimmune disease and as sclerosis multiple and rheumatoid arthritis. You can see here. So I propose that having a herpes in our body, inhibiting our DNases, is like having a permanent mutation. So if those enzymes, in theory, could degrade the virus DNA and they are being affected, they cannot degrade it and cause chronic diseases, which are diseases in unknown causes. And actually, there is a recommendation that people with rheumatic disease would do better if they are vaccinated with varicella zoster, which points to a link between chronic diseases and herpes viruses. And also, we got for that the vaccines, the varicella zoster vaccine, although they are not perfect. So some people got reactivation of varicella zoster after the vaccine, even if it's the subunit vaccine, not the attenuated virus that some children get. And there are even cases of fatal disease because of the vaccine. I'm not anti-vaccine at all, but I think we should do better vaccines than the ones we currently have. The only available one is for herpes zoster. All the other viruses, all the other herpeses do not have an available vaccine. So there is also an association between sclerosis multiple and maybe impaired DNases. So this is more evidence. And also, yeah, those are all the systemic lupus erythematosus that can be occurring due to inhibition by DNases. And in the case of cataracts, I don't see a crazy idea that herpes could be behind that the not neural degeneration, but this age degeneration, because it gets latent to the trigeminal ganglia, and during every stress it reactivates, and is possibly inhibiting the DNases that are required to form the crystalline of the eye. So this is only an hypothesis. I got some evidence myself. This is an experiment I did for a grant I got, where we got uninfected cells and then infected cells with the virus, and I saw that there is a decrease of DNases driven by the virus. So that means that the virus is targeting the DNases that we have that could degrade it. So with the grant I got, I proposed to find the viral proteins that are targeting our DNases, which are unknown, and also generate novel drugs which would release the DNases from the viral sequestration so they can do the function they are supposed to do in the cell, which is detect the foreign DNA and degrade it. So in that way, if we eliminate the virus, we can ameliorate diseases which we don't even know the cause. So it's good to do a research and know the cause of the diseases. In many cases, it's going to take a lot of years. But if we don't have a cure for that virus, like in the case of multiple sclerosis, we are in a catch. We cannot really do anything about it. So I'm almost done. This is just to throw the discussion. So this is a study of very stressed people. Those are astronauts, where they had their fluids collected to see which viruses they had. And they could find a lot of like herpes in the skin, of course, then Epstein-Barr in the blood, Epstein-Barr and herpes simplex and zoster in the saliva, as well as cytomegalovirus in the urine. Because of their accumulation in body fluids, I propose whether they could be a cause for diseases related to age or inflammatory disease, autoimmune disease, as could be temporal arteritis, gallbladder stones, pancreatitis, overall aging if they are infecting and degrading our bone marrow, or even part of the urinary and reproductive organ problems like kidney stones, prostate cancer, or synovial fluid as arthritis. So many thanks for your attention. I'll be happy to take questions and have a discussion. Thank you. I can't hear you. I'm on mute.
Thank you, Mila. That was really interesting. I must say I wasn't so aware about the chronicity of the infection and the high prevalence across the community. I've got some comments. Let me see. First of all, do you want to stop sharing if that's okay, so we can see if people have any questions? I can't see any hands raised while everyone is gathering their thoughts. So you propose to, what's your kind of, how are you going to go about eliminating the viruses? You said the objective should be eliminating the viruses and not focusing on proteins. So how do you suggest, how do you go about doing that?
So I'm only gonna study the proteins that can eliminate the virus. So those are the DNases. So I'm gonna see which host DNases, which DNases we have that are interacting with the virus. So I would infect the cells and then, let's say, I don't know if people know about biology so much there, but we will use a technique called mass spectrometry, which would find the proteins interacting with viral proteins. And once we know those interaction pairs, like DNases with the viral proteins may interact with those, then we will model different drugs and see which ones can disrupt that interaction. So I'm not focusing on the host protein, I'm rather focusing on the viral protein or the viral host interaction. And in that way, I can release the DNAs and allow that to chop viral DNA. So
Okay.
yeah, that way.
Interesting. And did you have any thoughts about, do you need to do any clinical study to kind of give you missing
Yeah.
pieces of puzzle?
Yeah, that'll take, that's the plan until possibly retire if I get funding after funding. So first we need to validate the mechanism at molecular level, then in cells, then we have to possibly go through animals, possibly mice. And if we are sure of that mechanism, then we can test in humans. So first you recruit some volunteers, maybe people that is immunocompromised or that have a life-threatening condition that may volunteer for that, or otherwise maybe they will die or they'll be blind, something that is something that lasts possibility. If in those volunteer group the drug is working well, then bigger trials will be done with more people, and then a third trial with many people from different places. And when all of that will be approved, then the drug can be commercialized. It could be 20 years time if any of those things happen, but it's better to start it than non-starting. Yeah.
Yeah. You mentioned that study done with the astronauts
Yeah.
about how they were shredding viruses when they were seemingly under stress. Do you think there has been, at the start of the presentation, you mentioned the link or kind of between different viruses and some of the chronic diseases like multiple sclerosis was one of the things you mentioned. Has there been any studies to say that patients who develop these diseases have higher levels of, evidence of kind of virus being proliferated at the time of diagnosis?
So it's very unclear the literature there and which virus and depends on the disease. So there are some suggestions, hypotheses, theories, but there is no final proof, as in the case of multiple sclerosis. There was a huge study studying 10 million people over a really long lifespan. So they are sure of that, but as far as I know, there is no yet clear link. There's proposals, there are papers, single publications, but
Yeah.
yet not clear. Yeah.
Okay.
And in the case of astronauts, I like to remark that nowadays there is this ambition to go to outer space and send people. But if people is affected with herpes, and herpes are causing neonatal mortality, that could be a problem in bringing humans to the space because they have less rate of survival. So
That's
there is
very
a person.
interesting. Yeah. Katie, do you have a question?
Yeah. Hello, thank you. That was really fascinating. So this is really far from anything that I know about. So my questions are probably very stupid. But one thing I was wondering is, so your potential solution, is it a vaccination or a treatment?
A
Or
treatment.
you don't know yet?
So I will say a treatment because vaccination, the ones we use only are dedicated, so they is to make antibodies against a single protein. And many cases is the protein that is gonna help the virus to get into the cell. So the idea is to block that entrance of the virus. But as far as we all know, the virus always gets into the cell. And the moment you have the virus in the cell, the moment you get stressed, it's going to make more copies and you're going to have that reservoir in you. So the vaccines may reduce the frequency or the pain or the symptoms, but they will never eliminate the virus, as far as you know. We got varicella zoster vaccine, that's the only one we got, and that never clears off the virus. First of all, the vaccine is the virus itself. It's attenuated, except for the subunit vaccine. It also has some not clearing the virus completely. I don't think the vaccines are just now the solution to eliminate completely. I think we may need a drug or a combination of drugs that potentiates our own defenses, the defenses we have to degrade those exogenous DNA, and that will be just to let the body see that we have a pathogen so it can degrade it. Because I'm sure the virus has defenses that are counteracting that, so the body cannot degrade it completely. Yeah.
And is it possible, so in the case of the astronauts, so you can have multiple of these viruses at one time?
Yeah.
Yeah, so you could have
all of
them.
us. Yeah, everybody has them. Like, for example, if you've got chickenpox as a kid, you got herpes zoster already on you, and possibly will reactivate when you are older, causing shingles. If you ever got infected with herpes simplex, you may have or not have, depending on how you are, whether you are stressed or not, those cold sores in the mouth or in the nose or genital sores. Also, Epstein-Barr, I think pretty much everybody has it, is causing the mononucleosis. So that's why it's proposed as chronic fatigue. Some people have chronic fatigue, an infectious mononucleosis because of the Epstein-Barr. So there are many, many common, and also the 3 days fever one, which pretty much every newborn gets them. So we got not only one, but many of them. And I think if we would not have them, we will have better quality of life because we wouldn't have those little problems that sometimes are big problems and possibly are degenerating our body as a whole and aging us more, causing arthritis or causing other autoimmune diseases. That's my opinion.
Thank you.
It's just to that point, you said if you didn't have those viruses, we would have better quality of life. Has there been a large scale population study done to prove that you had to, must be very difficult, because if you say almost everyone has it, then where are you going to find your control group to see how much damage to health and well-being these viruses are causing?
So I would say the largest study will be the one for multiple sclerosis, where you grab the people before they get infected, you detect when they get infected, and then track whether they had a disease afterwards.
Okay.
If you do that at global scale with every virus, that could help to find diseases. But I imagine they cost a lot, a lot of resources. It's not impossible, but I'm sure it's the case. Yeah.
Okay.
Mm-hmm.
Does anybody have any final thoughts or comments or questions?
I don't have any other questions, but I found that really interesting. It's well out of the remit of what I do, but so sorry I've not asked any questions, but I find that really interesting. Thank you.
Yeah, I would agree. So thank you for that. So I'll just make two kind of housekeeping announcements. So if you are interested to talk about any of your ideas, no matter how wide or different it is to what we do, we'd be open for that. So please drop me an email. It's a very informal meeting, and I'll be very welcome to provide you a slot. And our next seminar is 24th of June. Dr. Luis Santos from the vet school is going to talk about role of simulation-based learning in vet school, and using sociological risks and psychological shocks for veterinary students. So that will be interesting. So thank you everyone for joining, and special thanks to Mila. I'm going to do a
thank you
round of applause, and see you all soon.
Thank you.